Welcome to video 3 in our series on driving the power of single-cell technologies forward to the clinic with ScaiVision. I'm Sarah Carl, Director of Data Science at Skylight. In the last video, I talked about ScaiVision's representation learning algorithm and how it works. Today, I'll focus on how this approach retains single-cell resolution throughout the analysis, and why this can lead to increased discovery power versus conventional techniques. I already showed this slide in the last video, but I want to revisit it now with a focus on the limitations of the cluster or cell type-based approach. Whether you start with an unsupervised clustering or apply a reference-based cell type annotation, you're introducing some bias into your analysis. Clustering results depend heavily on the parameters used, which can influence the number and granularity of the resulting clusters, as well as the downstream results. And as anyone who's done cell type annotation knows, the results of different methods can often disagree pretty significantly. Besides this, the quality of the reference datasets available has a huge influence on the results, with rare, less studied cell types, as well as cancer cells, for example, being particularly difficult to annotate well. On top of this bias, with these approaches, you're actually losing some of the resolution that the single-cell data inherently brings, because you're pooling cells within clusters or cell types. Again, to remind you how ScaiVision's approach is different, we don't require any clustering or annotation upfront. Rather, we assign every cell a label that corresponds to the endpoint of interest, and then subsample groups of individual cells from each label to create many multi-cell inputs, all of which are fed into the models. So the only things that matter to the model are the endpoint labels and the cell-wise gene expression values, and each cell has a chance to influence the model's predictions. Moreover, what I find really powerful with this approach is that after we have trained models to make predictions on a sample level, we can use them to go back and assign a score to every individual cell, which indicates how predictive that one cell is of a given endpoint. We do this by sampling cells one by one and feeding them into the pre-trained model. Now, instead of a matrix, we have a simple vector of input features per cell, and multiplying this vector by each set of filter weights gives one value indicating that cell's response to each filter. These values are fed through the same fully connected output layer to generate a probability score for each label class. Then we calculate the log difference between these probabilities, which gives a measure of how strongly predictive each cell is and in which direction. What's nice is that this metric incorporates information from all of the trained filters, so it can correctly assign high scores to cells from multiple different cell types or populations. To show an example, here is a single-cell dataset from leukemia patients, labeled as responder or non-responder. We trained models to predict response at baseline, and then scored individual cells for predictiveness. In this case, positive cell scores indicate non-response and are labeled here in orange. And only the top 10% most predictive cells for each label are plotted for clarity. On the top, you see cells that came from actual non-responder patients. And there are two main populations of highly scoring orange cells, one in the middle and one as a separate cluster on the left side. If you look at the bottom UMAP, which shows cells from responder patients, these cell populations are still there, but they don't score as highly, as shown by the lighter color. The same is true in the inverse for the blue cells, which are the most highly predictive of response. You can see multiple dark blue populations on the bottom plot, and while those general populations are still there in non-responders, they have a lower predictiveness score. This type of analysis can help us understand whether the signal that the models learn comes from cell populations that are only present in samples from one label class, so indicative of differential abundance, or whether it comes from cell populations that are present in both classes, as is the case here, but which may have transcriptional differences, which cause them to activate the network filters more strongly. It could also help us understand why certain samples are predicted in a certain way, whether that's correctly or incorrectly, by pinpointing which cells within those samples gave the strongest signal in either direction. I'll use another example to illustrate how this unbiased approach can yield greater discovery power. We analyzed a publicly available dataset of CAR T cell therapy products, where the endpoints were high versus low-grade toxicity experienced by patients. In the paper, the authors used a standard unsupervised clustering approach and then performed differential expression analysis for each cluster for high versus low-grade toxicity. They identified one outlier population of cells that were associated with high-grade toxicity, which they called IACs. But these cells alone could not fully stratify patients by toxicity grade. Moreover, the cluster-based approach did not allow the authors to find any significant differences across toxicity grade in any of the main T-cell populations. On the other hand, by training models with ScaiVision and then scoring cells for predictiveness of high-grade toxicity, we recovered the IACs, but we also found additional populations of cells that were predictive, including within both CD4 and CD8 T cells. Summing all of these predictive cells together actually did allow for a perfect stratification of patients by toxicity grade, in addition to generating new hypotheses about the biological drivers of toxicity, which we could then delve deeper into by looking at the selected cells' transcriptional patterns in more detail. So this approach, retaining single-cell resolution, allowed us to both discover something new in the data that the traditional approach had missed, and to really pinpoint those most important cells in an unbiased way. In the next video, I'll focus on how we use explainable AI to prioritize gene or protein features for assay prototyping, thereby enabling translatability into the clinic. I hope you'll join me.